So we're going to take a step back with today's lecture and mainly focus on methodology. So remember that though the vast majority of this class is going to focus on statistics, statistical techniques, the appropriateness of statistics, and the assumptions those statistics need us to check before we use them and report them, that they're actually basically the top of a very large series of events that are more methodological. The first one, of course, is the very base, your research questions, your hypotheses. What are you trying to predict? What are you trying to identify? And what relationships are you looking for? So before we even move forward with anything else in methodology, we need to make sure that we have a clear understanding of what the question is we're asking and what are the specific testable hypotheses that we are proposing. And that's generally why in a research paper we often see such a large introduction section. It's a review of the literature, it's a review of what has been done in the past to help us guide ourselves to making a good research question, to asking good hypotheses that are testable in a way that leads us to our methodology. Whenever you're writing an introduction section, by the end of that section, pretty much your reader should be able to guess or almost write out your hypotheses. That's the goal of the section, is that you've basically created a document which makes such a strong argument for the questions you're going to ask that any reader reading it would come up with those questions as well. And those questions, again, need to be testable. In other words, they need to be stated in such a way that they could be supported or not supported. But they're also going to be stated in such a way that basically informs us what are the constructs, psychological constructs you're interested in looking at, and which ones are predictors and which ones are outcomes. And what is the relationship or the direction of the relationship you're looking for, and that kind of ties into last week's lecture looking at one-tailed versus two-tailed, directional or non-directional tests. Once you have your hypotheses, we're now going to actually get to the research methods itself. So the research methods is actually designing the study. What exact methodology are you going to use? How are you going to measure and manipulate or quantify your predictors, your IVs, your independent variables? And how are you going to manipulate or more specifically measure your outcome variable? What are the specific operations that you're going to do to create a study which can either support or refute your hypotheses? And as a part of this process is the quantification of those relationships, measurement. How do we actually define each variable in our study, each independent variable, predictor, and each dependent variable, outcome? And depending on these layers of the pyramid that we are going upwards, at the measurement phase, depending if those IVs and DVs are categorical, continuous, whether they're ordinal, all of those things we talked about last week start to define what statistics we can use. So once we know what we're measuring, how we're measuring it, and what relationships we're looking for, we can use that information to then determine what is the actual appropriate statistics to look for that relationship. And again, all statistics are going to be doing is specifically asking, is the relationship or differences you found between those independent variables and dependent variables more likely due to chance or more likely due to a fact that there's an actual effect there worth discussing and worth understanding? So based on this pyramid, we're going to be talking about the experimental method and then quasi or non-experimental methods in statistics. So again, we're not talking about statistics today. We're talking about the base of the pyramid. And the first thing we're going to be talking about is the ideal kind of gold standard in any kind of methodological study, and that is the true experiment. And we're going to be talking about how we create a true experiment, what are the requirements, and again, we get back to last week's lecture of Mills' canons, that there has to be a statistical relationship, the cause has to precede the effect, and we have to rule out any other potential cause or confound. So a part of the principle of experimental methodology is that we have to be manipulating the independent variable. We have to directly be changing one variable from one level to another. It can't be pre-existing. It can't be a pre-existing difference. It has to be something that we have taken control of. And generally, it's also something that we're going to be using random assignment to assign people to those different levels of that independent variable. In other words, we've taken control of the IV so that we can make that temporal precedence argument, that the IV did occur before the DV, the predictor did occur before the actual outcome, and we know it because we're the ones that controlled it. So we want to observe the effect of that manipulation, that control, if you will, on a second variable, a dependent variable, the IV. Yes, sorry, DV, dependent variable. So again, just the first question we have when we're talking about experimental methodology is simply the question of, can I control the independent variable? If it is something I can control, like group assignment, or exposure to an intervention or non-exposure to an intervention, or the presence of a drug versus the presence of a placebo. And again, I am using random assignment, which is getting to that confound issue, to assign people to those groups. If I have that, I probably can continue forth towards a true experiment, one that's actually getting at Mill's canons of causation. However, if what I'm interested in is not directly changeable by me, if I'm interested in gender differences, if I'm interested in differences between first-year workers versus senior workers that have been at a company for 20 years, if I'm interested in first graders versus second graders, all of those are pre-existing groups, pre-existing conditions that bring in confounds with them. That doesn't mean that it's not possible to do studies. In fact, a lot of our studies are non-true experimental because they're interesting things that we want to know more about. Just recognize that if that IV is something that is pre-existing, I can't take control of it, I can't ethically assign people to it, I can't get to true experimental. And again, the most basic form of the true experiment is a single IV with two levels with a single DV, generally going to be continuous or ratio data. So we have a categorical IV, groups, that we're assigning people to. So for example, this is a simple cognitive study on memory, words recalled. So our IV is the level of processing. In other words, when people are given the list of words, they're either given the list of words specifically in high meaning sentences. In other words, each word they're supposed to remember, they're given basically the definition of that word. Or in a medium level of processing. They're not given the definition, they're simply given the word with another word that rhymes with it that they're not necessarily supposed to remember. We then wait for a few minutes to pass, and then we have them recall the words. And then we look for the mean difference. We find, for example, that the high meaning group on average recalled 8 of the 10 words, while the medium or rhyming condition only recalled 5 of the 10 words. We would then run our statistical analysis. But again, if we have a group of people that we are randomly assigning to either the high meaning or the medium meaning group in one experimental condition, at one time, in one room, all of them exposed to the same word list, the only thing we've changed is whether the level of processing is high or medium, and then they all have the same outcome, the number of words they correctly recall. This is an example of a true experiment. We've manipulated the IV, we have used random assignment to remove possible differences between the individuals in the various groups, as each person had an equal chance of being in either group. We've made sure that there was no difference in time, no difference in location, and no difference in the operations that were done, which gets us to something that we can probably talk about, that if we find this difference is statistically significant, it's a meaningful difference, that we can say level of processing causes an increase in the words recalled. Higher processing results in more words being recalled. But recognize also that, just briefly, the limitations of this study. It is highly artificial. Everyone in this study knows that they're in a study. They're being actually assigned to different conditions. It's fairly clear what the study is going to be about, which means there are also maybe motivations. Now again, hopefully they're randomized between the two groups as far as whether people are wanting to help you with your study or not. So for example, that's what we often refer to as demand characteristics. So if, for example, I know I'm in the medium group, the rhyming group, and I know that that's the group that you expect to not do as well, I might decide I want to help you out, so I purposely don't try hard. Likewise, if I'm in the high meaning group and I know I'm in the group that you expect to do well, I might try to do a little bit better, especially if I want to help you out with your study. That's why we often want to set these studies up in a way that is called single or double blind, to make sure that our participants don't actually know what categories they're in. Again, it gets rid of yet another possible explanation for the results that you find. So that method of manipulation, generally speaking, we need to create at least two groups that differ as much as possible on the IV. And again, last week we talked about this a little bit in terms of power. The bigger the difference between your groups, the more likely you are to find a difference in your actual DV or your outcome. So things we can look at is control groups, experimental groups, and possibly placebo conditions. So a control group simply means they're getting nothing. They're the counterfactual. They have no intervention. Now, the problem with a pure control group, however, is that that does lead to a potential confound. So for example, if my control group does absolutely nothing and then just fills out a DV, an outcome, but my experimental group gets a 10-minute interaction video about studying. Now, my argument is that the content of that 10 minutes is what's causing the difference, that they're learning more about how to study and therefore they do better on the outcome. But what if it could be simply that that video actually increases their attention a bit? It focuses them. It has nothing to do with the content. It simply has to be doing with 10 minutes of basically something they had to engage with that the other group did not have to engage with. So just basically the mental awareness, if you will, or focus that that might cause. So it still gives me some information that this intervention is causing a difference, but what part of the intervention is causing that difference? And that's why we often may try to do a placebo group as well, or in addition, or maybe even replacing the control group. A placebo is no actual treatment, but treated procedurally similar to the experiment. So again, I have a 10-minute presentation on study style. So one way I could do the experiment is randomly assign people to either get the treatment, spend the 10 minutes, and then take the outcome test, or not get the treatment and immediately take the outcome test. I could also create a placebo group that gets a 10-minute presentation of equal length, equal number of slides, and kind of equal engagement, but on something completely irrelevant to the study. It's not about study techniques. Maybe it's about the mating habits of blue whales. That would become a placebo group. So experimentally, they were treated very similarly, but the intervention I was actually interested in, the content of the training, is now what can be tested. Did that content above and beyond just a 10-minute engagement video result in the effect? So obviously the best way to do this is to have all three groups, have a control group, have an experimental group, and have a placebo group. Again, this may be not possible due to time constraints, monetary constraints, or even participant constraints, but it's things to think about. Can I get a control versus experimental? If it's a pure control group, is that actually what I want, or would I rather have a placebo group? All of these are methodological questions. None of them necessarily directly affect statistics other than possibly changing the number of levels in your categorical IV, but they do have a fairly large impact in how you design your study, and how you design your study also then predicts what statistics you use. So again, the goal here is control. We're wanting to prevent, especially if we're doing a true experiment, any other extraneous variables from changing in the experimental process. That includes any environmental factors. So if you have to do different sessions with different participants in different categories in different rooms, different times, different places, those are all environmental factors. Also individual differences. So people differ greatly, and one of the arguments that can often be made about any experiment is that maybe the group that did better actually were better students or better studiers. It's one of the reasons that we fall back heavily on the concept of random assignment, that every individual coming into a study has an equal chance to end up in any condition. Now, this doesn't perfectly remove individual differences, but the larger your sample sizes, the more and more easily it is to argue that there probably is not individual differences because each group was randomly created out of your larger participant pool. So any extraneous variable, often referred to as a confound, provides an alternate explanation for the relationship between the IV and DV. It basically is giving someone the ability to play devil's advocate and say, I see what you did here, I see the study you ran, but you didn't account for this. Now, sometimes we'll actually discuss these extraneous variables, especially in a non-experimental study, as potential limitations of the study. So again, it's not that our goal is to always completely remove any possible extraneous variable. If that was our goal, we would only run experimental method sessions, true experiments in lab settings, which also have the disadvantage of being fairly artificial. So just recognize that if we can do a true experiment, we usually want to. But a lot of times we can't. But if we can't, we also want to play devil's advocate and look for those extraneous variables and at least discuss them or point them out honestly and legitimately as a part of the discussion of any paper. So we're going to talk a little bit about true experimental and quasi-experimental designs, and specifically what we're talking about is the variables of the design itself. So not IVs and DVs, but what are the components of experimental design, and how can you use them to potentially make better answers or to basically make a stronger argument that the effect you found was there when you can't use control groups or placebos. So we're going to talk about first an experimental design using these techniques, but then we're going to spend the rest of the lecture kind of talking about how we can use these components of experimental design to create better studies. Again, assuming we have enough participants, enough resources, and enough time to do so. So what are these components? The elements of design. The first are the measures, specifically observations. We'll usually refer to these as O's for observation. So these are our DVs, these are our outcomes. And again, a lot of studies will be very simple with a single measurement of the dependent variable, the O, but we can use additional observations and additional types of observations to make a more complex study. The next thing is treatments. So when are we interacting with our participants in such a way to add or subtract various treatments or independent variables? And again, these are going to be referred to as X's. So X refers to the addition of a treatment. The groups, how many groups are in the study, are the actual lines in our notation. So if we had a single group design, we would only have one line of X's and O's. If we had two groups, we'd have two lines of X's and O's. Three groups, three. So again, if your IV has 3 levels, you would be actually showing 3 lines, but specifically showing at what point do certain groups actually get exposed to your treatment. Finally, we're going to start each line with either an N or an R to represent random versus non-random. And finally, as we move left to right, that refers to the time. So for example, if I had an O, an X, and an O in a single line, that would be a single group treatment design. I would have a pretest because the first thing I do is that first O, I make an observation. I then insert the treatment and then I do the second observation. So again, I don't have a control group, but at least I have a pretest measure of what did the group look like prior to the intervention, then the intervention, and then what did the group look like on our outcome after the intervention. And again, since there would be no random assignment as there's a single group here, I would have an N at the start of that line. So it would actually read from left to right, N, O, X, O. So we're now going to talk about some of the designs we can use using these components, measures, treatments, groups, and assignment, and time, to make a better study that can actually get at hopefully a little bit better understanding of what we're actually interested in looking at. So this is that very simple random assignment, true experiment that I talked about earlier, but also with a pre-test and a post-test. So we've got 2 lines, that's group 1 and group 2. Group 1, the top line, we have R, random assignment, and at that point, from that random assignment, we actually have a first observation, a pre-test. That pre-test is then followed by the actual adding of the treatment, the X, followed by a post-test, observation 2. So again, the top line, half of my participants at random are assigned to that group. They're pre-tested, they get the treatment, they're post-tested. The second line is my control group. So in that case, again, randomly assigned to this group, an equal chance to end up in the top line or the bottom line. They're observed the first time, they do the pre-test, and then they have no treatment. This is a true control, followed by a second observation. And again, going from left to right is the time of the study. First random assignment, then the pre-test, then treatment or not treatment, and then post-test. And again, if we found that the top group actually had an improvement in their second observation, but the bottom group did not, we have a pretty strong argument, if that's a statistically significant relationship, that this was a causal event.